All right, in this video, we are going to take a look at the major methods that have been used to sequence DNA. And we're going to start out with a more historic method, and then we'll work our way all the way up to the most contemporary methods that are used to sequence DNA. Along with that, we're also going to talk about the process of gel electrophoresis, which is a process that's been harnessed to help with sequencing DNA. So basically, we're going to talk about how you wind up getting something that looks a lot like what's pictured here on the slide, which is actually a sequence of some DNA. And so basically, what DNA sequencing technology does is it uses what we know about the process of DNA replication and harnesses that to read the genetic code. So we know exactly what the sequence of A, T, C, and Gs are in whatever organism is of interest. So this is a very brief history of DNA sequencing. So this method was first developed back in 1977 by Frederick Sanger, who came up with a technique that used what are called chain-terminating inhibitors, which we'll talk about more in a moment, and radioactive probes to start sequencing DNA. By 1987, that technique was combined with another process referred to as capillary sequencing. Instead of using radioactive probes, there was also a switch over to using fluorescent labels, much safer. And so that technique then became known as automated Sanger sequencing. And that's the first major sequencing method that we're going to examine in detail. And this is a method that was used for the original Human Genome Project, so sequencing the first human genome. So then, between 1994 and 1998, a second method developed that's now referred to as next-generation sequencing. And next-gen sequencing only became available commercially in around the year 2005. And with this shift, it suddenly started to become possible to sequence many more genomes much more cheaply. And now we're at a point where there are at least 4 different DNA sequencing methods that are in wide use. And so we're going to explore one of these types of methods called single molecule sequencing, just to get a sense of the range of different techniques that are used to sequence DNA, because these different methods vary in how much they cost, they vary in the length of the sequence that can be generated, they also vary in how accurate they are, whether or not the technique is portable, and more. So it's useful to know about multiple methods just to have the perspective on different techniques that are available and what might be appropriate for a given situation. So here is a graph that shows the speed of sequencing through time. So back in the early days, using the gel-based methods, you notice the speed of sequencing was fairly slow. You could only get up to about 100 kilobases per day. And once first-generation sequencing took off, so that capillary sequencing process, suddenly, we went up in orders of magnitude in terms of how quickly we could sequence a given genome. Here's where Illumina, so the next-generation sequencing method, really started to take off. So again, there's a huge increase in our speed of sequencing. And now that we're into this next generation of sequencing, our sequencing speeds have just continued to rise dramatically. So it's now really, in a lot of ways, it's almost trivial to sequence a genome now, where it used to take a decade and billions of dollars to sequence a genome. And as kind of a side effect of the development of a lot of these molecular tools, it's actually now possible to get at least parts of your own genome sequenced. So there are companies such as 23andMe that are interested in helping you learn more about your own genotype and about variation in your genotype. I have this phrased as a question on this slide, should you? Because although it's possible to have your genome sequenced, there are a lot of concerns surrounding that decision as well. And so there's a lot of ethical issues that also come to the forefront when we talk about biotechnology. And I'm just going to touch a little bit on those ethical issues across these videos. And I'm not going to present any strong positions on them. I'm more just going to raise the questions as things for you to think about. Okay, so this the oldest sequencing method, Sanger sequencing, relies on a phenomenon referred to as dideoxy chain termination. And that sounds really fancy, but in practice it's not all that fancy, and I want to convince you of that. So here are the ingredients that are involved in Sanger sequencing. So first off, you need DNA, right? Whatever it is that you want to sequence, you need that organism's DNA. Then you also need a DNA primer. And so this is very much like the RNA primers that occur during DNA replication, except they're made out of DNA instead. And generally these are short sequences, so you see a sequence here that's 4 bases long. And so the chances of finding that sequence somewhere in a genome are pretty high. You also need some DNA polymerase, right? Because that's what elongates a piece of DNA. And then you need in your soup a collection of the deoxyribonucleotides. So those are the 4 bases that can be added to elongate a piece of DNA, right? And those are generally abbreviated DNTPs. And in addition to that, in this case, there are also dideoxyribonucleotides, so DDNTPs. And you'll notice these differ from the deoxyribonucleotides in that they're missing 2 oxygen groups. And the consequence of that is that when these get added onto a piece of DNA, it's no longer possible to keep building off of them. So they are, if you add these into your soup, then they're going to, anytime that they get added onto the end of an elongating piece of DNA, that elongation process is going to stop there, and where it stops is also going to be labeled with a fluorescent color. So you'll notice here that there are 4 different colors, fluorescent colors associated with each of the 4 different types of nucleotides. So you add that special type of dideoxyribonucleotide to your soup, and then you start to facilitate the process of DNA replication using the DNA polymerase that you've added and the DNA primer. And I'm going to talk a little bit more specifically about how that process gets going in a later video. But basically what will happen is your primer will bind, then DNA polymerase is going to start synthesizing new DNA, and based on the mixture that you have in your soup, most of the time it's just going to add regular nucleotides if you've got a whole bunch of these DNTPs. And then every once in a while, though, it's going to add one of these dideoxynucleotides instead. And so then for that round of DNA replication will come to an end, and you'll have a fluorescent label at that final position. And you'll notice here that that's going to be complementary to the nucleotide that is on the strand of DNA that you're interested in. And so if you let replication happen again and again and again, then basically you're going to wind up with a mixture, when this is done, of pieces of DNA that are different in length, and where the final nucleotide has a fluorescent label on it. Okay? All right, so then what? So then the next question is, well, is it possible to separate out these pieces of DNA of different lengths so that we know something about how long that piece is and then what color fluorescent label we get at that piece? And so this depends on another property of DNA, which is not just the size of the strand, but also the charge of a DNA strand. And so what you have to remember is that DNA as a molecule overall has a negative charge. So what you can do is you can take a mixture of DNA molecules of different sizes, as illustrated on this thread, or on this slide, and if you inject that mixture into a little well, into a gel. So the gel here is just some kind of matrix. It's very much like Jello. So, a substance like Jello, it's a mixture of materials that provide structure, but also aqueous space. And so because of that, it's possible for molecules to travel through this aqueous space. And if you apply a charge across this space, so if you have a positive voltage at the far end and then a negative voltage at the near end, since DNA is negatively charged, that DNA is then going to move towards the positive pole. And the smaller the DNA piece of DNA, the further it will move. And you can actually do this in a way that generates a set of bands. And so your smallest pieces will be down here, your largest pieces will be up here. And you can look at the color that shows up at each of these points. And that will tell you the sequence of bases, right? Because these are in order. So that'll tell you the sequence of bases in the DNA molecule of interest. So again, this process is called gel electrophoresis. So the electro means electrical, phoresis means transport. So gel electrophoresis, just sending a DNA molecule through a gel to separate out pieces of DNA by size and by charge. So the first major sequencing method I mentioned, Sanger sequencing, relies on this principle, and the only major innovation that developed was to, instead of running everything out on gels, which are big, expensive, slow, cumbersome, instead he figured out that you could run your DNA through a capillary column. So that's just a very thin glass column of liquid, and use that same method to separate molecules by size. And then as you run that DNA through the capillary column over time, it's kind of like if you took this gel and you just watched this one location. And if you keep running this current, stuff is going to keep traveling downwards. And so over time, what would happen is first the red point would go past, so you'd note red. Then the blue point is going to go past, so you'd note blue. Then the green point, so you got green, etc. And so you can, using this method, just keeping track of what goes past at what time at a specific point, you can then come up with a sequence that matches the sequence of DNA that you're working with. This is a little faster than gel electrophoresis, but as I note here, this is actually still a fairly slow process overall. So this method made it possible to start sequencing relatively long pieces of DNA, so up to about 450 base pairs long, but you could only run one sequence at a time, and you have to start out with a lot of DNA to get this method to work. And it's also very expensive to use this type of sequencing. Those dideoxynucleic acids are not cheap. So the total price for Sanger sequencing winds up coming out somewhere around 2 cents per base. And if you start looking up how large different genomes are, you're going to discover that's going to get really expensive in a hurry. So part of what that means is that people had a lot of motivation to figure out other methods to sequence DNA that were a lot cheaper and faster. So that brings us to the technique of what's now referred to as next generation sequencing, to contrast with Sanger sequencing. So for next generation sequencing, some of the principles are related, but the fundamental process is different. So this sequencing method starts with chopping up genomic DNA, and we'll talk some more about how that works a little bit later on. After you chop up the DNA or fragment it, then you isolate those fragments on little glass beads. And then once the fragments are isolated onto and trapped onto these beads, then you use a process of polymerase chain reaction, and we'll talk about that process in a later video, to make extra copies of those fragments. And then your beads have been prepared, and you're ready to put them into a well and a plate that will then contain DNA polymerase, and it will also contain more of those DNA primers that I showed earlier. And so with these plates you can generally run on, I think it's at least 196, if not more, samples per plate. So they'll hold a lot of different pieces of DNA in a single plate. And then what happens is these are set up with instruments that will then wash the whole plate with a solution that contains one of the 4 nucleotides. And so, for example, it'll first wash with A, then it will wash with T, then it will wash with G, and then it will wash with C. And then that whole process gets repeated over and over and over and over again. And I think these pictures are kind of hilarious because, yeah, a lot of this is just automated at this point. So you just kind of interact with giant machines if you're doing this type of work. So then what happens is in each of those wells, every time that a new nucleotide gets added on to one of those pieces of DNA, there's a flash of light that gets released. And so over time, so you've got this big plate, you're pouring on A, T, G, and C. So let's say that you add on C, and you might see, flashes of light across some subset of the wells. Then a computer is going to record, okay, so well A1, C, A4, C. Then, if you put in G next, then the computer is going to note, A2, G, etc. So then the computer is going to build up sequences associated with all of the wells that are being run simultaneously. Okay, so here's some of the benefits of next-gen sequencing. So first off, it is way cheaper than Sanger sequencing was. And you can fit a lot of samples onto one of these wells, so it's a very high throughput process. So you can sequence things relatively quickly using this method, especially compared to Sanger sequencing. And it's also a reasonably precise process. So the error rate is somewhere between 0.46 to 2%. So not perfect, but not bad, all things considered. There are some downsides to this method, which is you can only use this technique with short pieces of DNA. So this is referred to as a short read method. So the maximum sequence length you can typically read with this is a sequence that's about 400 bases long. And by itself, that's not necessarily a deal breaker. What winds up being a deal breaker is eventually, after you have sequenced pieces of DNA, you need to stitch those pieces back together to figure out, what's a chromosome, what's a gene. And the shorter the sequences that you have to work with, the harder it is to line them up and stitch them back together with each other. So that lining up, stitching back together process is referred to as assembly. So, there are some organisms that have genomes that have a lot of repetitive DNA in them, repetitive sequences. And so it would be very difficult to assemble the genome for an organism that has a lot of these repetitive sequences if all that you have are really short sequences to work with. So that brings us in to kind of one other contemporary method that's gaining quite a lot in popularity, which is called single molecule sequencing. So I'm going to talk about two single molecule sequencing methods. First is a method that's been trademarked by a biology company called Pacific Bio, so they call it smart sequencing. And for this technique, what they do is they have a method that allows them to attach a DNA polymerase at the bottom of a well. And then very similarly to what I showed for next-gen sequencing, every time this DNA polymerase adds a nucleotide, there's a flash of light. The benefit here is that if you have just a single DNA polymerase, you can start to sequence longer pieces of DNA using this method relative to what's involved in next-gen sequencing. So the other single molecule sequencing method that I'll talk about is called Oxford nanopore sequencing. And this actually relies on a different technological process. Basically, for this technique, pieces of DNA are passed through a very tiny protein nanopore. It's almost like the nuclear pores on the nucleus. And so also relying on the fact that DNA is charged, if you pass this DNA through one of these tiny protein nanopores, and you have very precise instrumentation to measure charge, you can measure voltage changes as that DNA molecule passes through the protein nanopore, and specific voltage patterns are associated with specific nucleotides. So here's an example of a readout using nanopore sequencing. So you notice on the x-axis here is time. So through time, here are the charges associated with the nucleotides passing through. So again, just one more way to sequence DNA. So these methods also have their pros and cons. An advantage is that you can get very long sequences using single molecule sequencing. So instead of somewhere around 400 bases to 450, now we're talking about a sequence that's somewhere between 5,000 to 8,000 bases long. And in some cases, you can even get sequences that are as long as 30,000 bases. These are also very accurate methods, so 99.99% accurate. And because you're working with just single molecules of DNA, you don't have to average across a big pool of DNA. So you can get sequences that are specific to every individual. One other really cool thing is that nanopore sequencing technology is also very portable. So here's an example of a nanopore sequencer, and it's just hooked up to a USB drive. So I have a colleague who's actually taken these to remote field locations, and he's been able to sequence things in that location just on the spot, which is pretty amazing. There are some downsides. These methods are still very expensive right now, so they're limited to special applications, and we don't entirely understand why, but in some cases, nanopore sequencers just don't work. And so, hopefully we'll be able to puzzle through that at some point, but there are still limitations to working with these sequencing methods. Okay, so here I've taken you through a tour of DNA sequencing methods from old school Sanger sequencing, how the whole process got started, through to next generation sequencing, which is still what I'd say is one of the predominant methods of sequencing that's used, up to single molecule sequencing, which has really taken off and again expanded our ability to sequence all kinds of genomes. But there's something else I haven't talked about yet across at least two of these sequencing methods, which is that two of these sequencing methods require a lot of DNA. And okay, that might not be a huge deal if you're studying a relatively large organism who's willing to contribute a relatively large bit of DNA. But it's also interesting to study smaller organisms that don't contain all that much DNA. So one of the other things biologists have had to do is figure out how can we get a lot of DNA. So that's something that I'm going to talk about in the next video.